Hi guys, so today I'm going to be talking about translation. So what translation is that it's the second stage of protein synthesis, where the first stage is transcription, where we are making the mRNA strand. So in translation, what happens is that we're using this mRNA strand and the sequence of codons found on this mRNA strand to assemble or to be translated into a specific sequence of amino acids. So we're basically reading this mRNA strand to form a specific sequence of amino acids. And this sequence of amino acids will form a chain called the polypeptide chain. And this will be further modified and changed into a protein. So this will be the overview of what happens during protein synthesis. And we have looked into what transcription is, which happens inside the nucleus, and we will be making the mRNA strand, the mature mRNA strand. And the mature mRNA strand will move outside to the cytoplasm through the nuclear pores over here. And the mature mRNA strand will then bind to a ribosome, where translation could later on occur. And we can see here that tRNA molecules will be what will be reading the mRNA strand and forming this amino acid chain at the end. So I will be talking about these molecules later on. So first, let's talk about ribosomes. A ribosome is basically an organelle where we can find in the cytoplasm. It's made out of two subunits. The first is the large subunit, which is right over here, this one, and it has two tRNA binding sites. So a tRNA molecule is what is needed to bring the amino acids in order to make this chain to the ribosome. So at any one moment in time, the large subunit may have two tRNAs binded towards it. And then we have the second small subunit. And this small subunit will be what binds to the mRNA strand. So now let's talk about what a tRNA molecule really is. So tRNA molecules are basically transfer RNA molecules. And from the name itself, you can see that it means that it's transferring amino acids to the ribosome during protein synthesis. And they are all found in the cytoplasm. So a tRNA molecule will have this amino acid binding site at the end of the molecule. And it will be bringing a specific amino acid which corresponds to the anticodon that the tRNA molecule has. So what the anticodon is basically a triplet-based sequence of nucleotides which is complementary to the mRNA strand. So if our mRNA strand will have GCC, and since according to the concept of complementary base pairings, we have A pairing up with U and C pairing up with G, then we can see that the anticodon that would correspond to GCC on an mRNA strand would be CGG. And that would be the anticodon that would pair up with this codon on the mRNA. And this tRNA molecule will hold a specific amino acid which corresponds to the CGG anticodon. Okay, so now let's jump into the process of translation. The first stage of translation will be the initiation stage. What happens here is that we have an mRNA strand attaching itself to the small subunit of a ribosome, which will be this one. It will always attach itself at the start codon, which is usually AUG. This will be the nucleotide base sequence that will usually be the start codon of an mRNA strand, as you can see in the diagram. So then a tRNA molecule can bind towards this start codon if it has the complementary anticodon that would correspond to AUG, which in this case, it would be UAC. So the tRNA molecule with this anticodon would approach this AUG start codon and pairs up with it according to the complementary base pairing concept. And now we have a codon and an anticodon standing next to each other. And what would form in order to kind of bind these together is a temporary hydrogen bond. So temporary hydrogen bonds will form between UAC and AUG to keep the tRNA molecule and the mRNA strand together. And now we have this complex, as you can see in the diagram. The tRNA molecule with the start codon, which corresponds to the start codon, will always bring the amino acid methionine. So methionine will always be the first amino acid in any polypeptide chain that we are trying to make. So then what happens next is that a large ribosomal subunit will bind to this tRNA molecule, because remember, as I've said here, a ribosome at the larger subunit will have 2 tRNA binding sites. And that's where this tRNA molecule will bind to, and therefore constructing the full ribosome. And now we can move on to step 2, where a second tRNA molecule can bind. Because remember, I've said that there are 2 tRNA binding sites of the larger ribosomal subunit. So a second tRNA can bind at its binding site, and this will be always according to the complementary base pairing concept. So this second tRNA molecule must have the anticodon that is complementary to the codon of the mRNA strand, just like the process of the first tRNA molecule binding. And it will form temporary hydrogen bonds. And as you can see, it will be bringing the second amino acid, which will be added to the amino acid chain later on.